Hello everyone, welcome back. Today I'm here with this 10th part of this questionnaire. Today I will discuss some questions from plant embryology and plant anatomy. Let's start. The first question is Jeffrey reported cleavage polyembryony. In our options are Erythronium americanum, Nymphia albina, Eulophia epidendria, which is an orchid, then Allium odorum. So its right answer is एरिथ्रोनियम अमेरिकैनम. सो इसमें सबसे पहले जेफरी ने 1895 में सबसे पहले ऑब्जर्व किया था polyembryony को. This is the plant Erythronium americanum, which is commonly known as yellow trout lily, and this polyembryony was first observed by Leeuwenhoek in orange or citrus fruit. So Jeffrey in 1895 described cleavage polyembryony in Erythronium americanum, where the synergids disintegrate soon after the fertilization, and zygote divides to give rise to a small group of cells irregularly arranged. Later 2 to 4 outgrowths develop from the lower end of this group of cells, and each developing into an independent embryo. So that is called polyembryony. So it was firstly observed by Jeffrey in Erythronium americanum. Then this cleavage polyembryony is more frequent in Orchidaceae also. So this plant that is called Eulophia epidendria. Yeah, so here in this plant it was observed by Swami in 1943. Next is Allium odorum. So it may be cited as an interesting example where polyembryony occurs due to operation of more than one methods described above, and at the same time in this plant a total 5 embryos develops. That is 1 from zygote, 1 from synergid, 2 from antipodals, and 1 from integument. So this is how these 5 embryos are. Next question, in gymnosperms the endosperm is, our options are haploid, diploid, triploid, or tetraploid. Its right answer is haploid, and this is formed prior to fertilization. Fertilization से पहले ये बन जाता है, and it is haploid. While in case of angiosperm, this endosperm is triploid because it is formed after fertilization. Here the 2 polar nuclei fuses with 1 male gamete and results in formation of this triploid endosperm, which is formed after fertilization. So this is evolutionary advanced step. So in case of gymnosperm, this endosperm or this nutritive tissue is haploid and is formed prior to fertilization, while in case of angiosperm this is triploid and is formed after fertilization. So the endosperm of gymnosperm is haploid, and it is pre-fertile. It is a pre-fertilization tissue and is equivalent to female gametophyte, hence it is haploid in nature. But the angiosperm, it is post-fertilization tissue, fertilization के बाद बनता है, and is generally triploid in nature. This is triploid. So this is important difference between the endosperm of gymnosperm and angiosperms. Next question is, when pollination takes place between the flowers born on 2 different plants, the process is called, our options are geitonogamy, xenogamy, hybridism, this is hybridism, and monogamy. So its right answer is xenogamy. Xeno means alien. So the plant of the same species which are present on different locations, pollen grain from that plant enters the stigmatic surface of the other plant of the same species. That is called xenogamy. So there are the differences: autogamy, geitonogamy, and xenogamy. Autogamy, it is the transfer of pollen grain from the anther to the stigma of the same flower, while geitonogamy transfer pollen grains from the anther to stigma of another flower of same plant. So plant is same but flowers are different, while xenogamy transfer pollen grains from anther to stigma of another flower on the different plants belonging to same species. So this is the question, this is the answer of this question. Here autogamy does not lead to genetic variation in progeny. Again, geitonogamy, it does not lead to genetic variation, while this xenogamy brings variation. Here autogamy, there is no need of any pollinating agents, and in case of geitonogamy, pollinating agent is needed, and xenogamy, it requires an external agent for pollination purpose. This autogamy occurs in clistogamous flowers which are closed flowers, while geitonogamy, pollination occurs in chasmogamous flower, means these flowers are open. Then pollination in chasmogamous flower, again in case of xenogamy. Next is hybridization or hybridism. Here this is artificial method of pollination which is used to bring some superior cultivars. So hybridization is controlled method of pollination. It is under the control of the will of a scientist, So hybridization is a controlled method of pollination in which the pollen of two different species or varieties is crossed by human intervention. So that is called hybridism. The next comes monogamy. Monogamy means here the plant species produces pollen and diffuses with only single plant species or same plant species once in a lifetime. So this is the photograph which shows different events of pollination, that is self-pollination, that is called autogamy, and cross-pollination between two different flowers of the two different plants of same species, and heterogamy, that is the pollination between the two different. Next question is, development of gametophyte from a cell of sporophyte without intervention of spores is known as. Options are apospory, apogamy, diplospory, and sporogamy. I have already discussed this question in previous part. So its right answer is apospory. Apo means without, spory means spore formation. So it is a formation of gametophyte from sporophyte directly without the formation of spores. Here the gametophyte found is diploid. और यहाँ पे जो gametophyte बनता है that is diploid. So we have difference here between apospory and apogamy. So apospory is formation of diploid gametophyte without meiosis and spore formation from the vegetative non-reproductive cell of sporophyte. So directly sporophyte से gametophyte बनता है without meiosis. While in case of apogamy, it is a type of production which occurs in some ferns in which the sporophyte develops from gametophyte without the fusion of gametes. That is a reversal of this apospory. The next is production of gametophyte without production of spore in case of apospory, while apogamy, it involves formation of embryo without undergoing fertilization. This apospory produces a diploid gametophyte and this apogamy produces the haploid embryo. It occurs in anthoceros, that is apospory, and this apogamy occurs in funaria. Next, we have different terms, that is sporogony and schizogony. Sporogony, this term is used in case of some parasites like mosquito. So sporogony is an asexual production that produces sporozoid, while schizogony is an asexual production method where sporozoites increase in number upon division. This sporogony produces sporozoites using gametocytes, while this schizogony multiply the sporozoites. Here this method, schizogony, multiplies the sporozoites inside the human host. This sporogony involves gametocytes. It does not involve this schizogony does not involve gametocyte. Sporogony is exogenous process, while this schizogony is endogenous process. This sporogony requires vector, means it occurs in the vector, and this schizogony, it occurs in the human cell host. Next thing is apomixis. So apomixis means it is the formation of the seed without fertilization. So there are different types of apomixis. Generally diplospory, apospory, adventitious embryony. So firstly diplospory. Here the embryo sac is derived from megaspore mother cell, and that is derived directly by meiotic division, और इसमें कोई भी meiotic division नहीं होता megaspore mother cell में. And this is formed due to default, or we can say abnormal meiosis. So meiosis होता नहीं है. In the meiotic diplospory, the megaspore mother cell divides meiotically thrice to form 8 nuclei, and embryo sac is unreduced and have same number of chromosomes as the genetic material. Next is apospory. Apospory, here the new cellular cells give rise to apomictic embryo sac. It is the most common type of apomixis in higher plants. Here aposporous initial cells differentiate and undergo mitosis to produce an embryo sac. Sometimes multiple embryo sacs may be found. The initiation of apospory embryo sac and sexual one can occur simultaneously. It can displace the or interrupt sexual embryo sac formation. Next is adventive embryony. So it is a type of sporophytic apomixis where embryo are produced directly from new cells or the integument of it develops by the meiotic division and forms a bud-like structure. Simultaneous fertilization in adjoining sexual embryo sac is required to form viable seed. Developing embryo goes towards it to obtain nutrients and signal from the embryo sac. Next question is, history theory was introduced by our options are Schmidt, Henstein, Nageli, or Schmidt. So his right answer is Henstein. This theory was proposed by Henstein. So firstly I will discuss about it is all the theories of shoot, apical, meristem development. So there are three different theories. First is apical cell theory that was given by Nageli, then histogen theory proposed by Henstein, where there are different layers of tissue which form different parts, like we have dermatogen that will form the epidermal portion, then Perivallum, that we found the cortex portion. Pleurome, that we found the stellar system. Next, tunica corpus theory, it was proposed by Schmidt in 1924. Here we have the two layers. Outer one is tunica, which is formed by one or two layers of cell that will give rise to the outer dermal tissue epidermis, then corpus, which is a central cell formed of cells which divide in all the planes. And in case of root apical meristem, there is equivalent theory which is just similar to that of tunica corpus theory, that is called corpor cape theory. So that is applicable in case of root, that was proposed by Schmidt. Firstly, apical cell theory, this was proposed by Nageli in 1944. According to this theory, the apical meristem consists of a single apical cell, and this cell is interpreted as the structure and functional unit of apical meristem. So the cell is very large and is shaped like an inverted pyramid. So this is applicable for lower plants. The lower plants only के लिए applicable है ये theory. Next theory is the histogen theory, which was given by Einstein in 1868. So this theory, according to this theory, a plant or body originates from a mass of meristem where the following histogens or regions can be distinguished. First is dermatogen. Derma means skin, that is the outermost layer of meristem, and it gives rise to the epidermis of root and stem. Next is peribellum, it means clothing. So region occurs internal to dermatogen but peripheral to pleurome. So histogen is destined to form cortex of root and shoot and inner tissue of leaves that surrounds the pleurome. The next is pleurome. Pleurome, it means that the thing which fills the central region. So this region gives rise to vascular cylinder, basically stellar system जो बनता है vascular system that is formed by pleurome in case of stem and root, including pith. So it is the central core of root and shoot, and the cell composing the zone are very irregular. The region is enveloped by a variable number of mantle-like layers, which are represented by dermatogen and peribellum. Next theory is the corpor cape theory, which is applicable in case of root apical meristem. So here the cells in root apex divides in a T-shaped pattern. This is very important, T-shaped pattern, and outer part of the root apex is called cape, and the inner part, the central part, is known as corpus. So cape and the cells here divide horizontally first, and the lower cell divides longitudinally and give rise to the structure which is T-shaped. This theory was proposed by Schmidt in 1970. The theory is comparable to the tunica corpus theory of shoot apical meristem. The tunica corpus theory in case of shoot apical meristem given by Schmidt in 1924. It is postulated that tunica corpus theory. This was all about for our today's discussion. Hope you will get some idea from this presentation. Thank you. Have a great day.